audible. Um, Please let me know if the screen is not visible at any point of time. Somebody was facing difficulty with it last week. Okay, so just let me know if there is any trouble. So, good evening. My name is Ankita Bhattacharya. I'm your TA for this course. As you already are familiar, we have been having some discussion sessions for the past few weeks, where we take up the topics that were discussed in the video lectures uploaded for the course, and we get into some extra detail about them. We revise some of the core topics so that we can refresh if there are any areas that are a little confusing or that need more clarification. Apart from that, we go through some questions, which especially the questions that were part of the previous week's assignment, and we try to address those for our better understanding. So this week, I have not included fresh sample questions because most of them were very overlapping with the ones that we've already discussed in the last week. If you want, I can include them in the upcoming class. But just to recap what has been discussed in this week. So the idea mainly centered around the structure of antibodies, the way in which isotype switch is carried out, that is switching from one type of antibody to another type, the mechanism, the molecular mechanism that operates in that case. And also following that, the idea of major histocompatibility complex or MHC, what is the need for it? What contribution does it have to the immune system and overall defense of any individual? What constitutes the MHC and how is it relevant in our case in particular? So before we get started again, just to iterate, please interrupt me at any point of time if you have any questions, if anything I said was not clear, or especially if you can't hear me or cannot see the slides. So with that in mind, we'll first start off with a discussion of the questions that were given as the assignment for week 3, which you have already submitted. Um, so the first question is about what is the minimum number of disulfide bonds that are required to maintain the structure of an antibody. So what you need to keep in mind here is that they're asking for the minimum number and not for what is the actual number of disulfide bonds. So it's very easy to get questions like this wrong. You need to be just a little mindful about it. So if you remember, we had talked about Yeah, so we had talked about the structure of antibodies previously. So you remember that there is one heavy chain which is joined with another heavy chain, and there are two light chains which are placed in very close proximity. And the disulfide bonds, they exist between the heavy light chains individually and then between the two dimers. So it was called as the dimer of dimers, thereby highlighting the fact that there are four, a minimum of, not minimum, but usually four disulfide bonds that help keep the structure intact. But if we are to ask what is the minimum number that is required, then it would simply be 3. You just need to join the heavy and light chains for each dimer. So just to show it by a different color, just the presence of a disulfide, yeah, just the presence of a disulfide bond here and another disulfide bond there, and finally another I think the pen's working. But anyway, you get the point that one disulfide bond between these two, another disulfide bond between the other variable and heavy chain, and another one between the two heavy chains would be enough. So 3 would be the answer in that case. Let me just erase this because it's not very clear. Yeah. So that was the answer to the first question. Second was about the major classes of light chain constant region sequences. Okay, before we proceed further, you can maybe refer to Kubi or any reference book that you have been looking at till now to see what is the exact number of disulfide bonds that are present in each type of antibody. We will be getting into it, but just for your own reference, you can go through that as well. The second question was about the major classes of light chains. Now, this is obviously a very factual question. We looked at the genomic arrangement of different types of antibodies, and we saw that there are two types of light chains, the kappa and the lambda light chains, which form combinations with all of the different heavy chain genes. And in combination, they help in increasing the diversity of antibodies, which is further helped by somatic hypernucleation, p-nucleotide and nucleotide addition, and so on and so forth, junctional diversity, so on and so forth. We have covered all of this in the previous class. In case you have not been able to go back and look at that, please do so. The slides are uploaded. And if you are not able to access the slides, please let me know. I will speak to somebody who is in charge of the course so that you can get in touch with them. The third question was about papain digestion of immunoglobulin G or IgG. Yields what type of fragment? So if let me just go back to that slide. So easier. Yeah. So this is the structure of the antibody that we were talking about. Here are the disulfide bonds that we talked about. That usually there are 4, but 3 would be minimum. If we talk about the cleavage site of papain, then it cuts right here, somewhere above the hinge, whereas pepsin it cuts between or below the hinge. So their cut sites are different, and therefore the types of antibody fragments that would be generated in each of these cases is also different. So since the question asks us about papain digestion, the resultant fragments that we will get would be 2 Fab fragments and the FC fragment, which would be joined to each other because the disulfide bond would still be interlinked. If the enzyme was different, the type of fragments generated would also be different in that case. Next was about hypervariable regions in an antibody light chain. What are they comprised of? They are comprised of alpha helices, beta sheets, loops, or none of the above. This we did not directly take up in the discussion session, but if you remember the lectures by Professor Steve, then you would remember that the loop regions which are present, like if you look at the structure of antibodies, it is made up of alpha helices present on two sides with beta sheets interspersing it, but it is actually the loop regions which are present at CDR1, CDR2, and CDR3, which are the hypervariable regions, and they are very easy to or very prone to getting mutated, as a result of which there is a huge diversity which can come about. And therefore, the answer to this question was about the loops, which are forming the hypervariable regions. Next question was about the expression of which of this following isotypes occurs by class switching, mu, delta, gamma, or all of the above. So if you remember the arrangement, that the first antibodies that come about are the IgM and IgD, which are arranged. Again, let's just quickly go back here. So if you remember this arrangement here, you can see that C mu and C delta are the first subtype which are generated as a result of VDJ recombination. And from there, if there is a need for it, or if the type of immune system or immune response that has come about requires some other antibody, then there is an isotype switch. And under those circumstances, these different types of antibodies would be transcribed and translated. So the first two are IgM and IgD. Therefore, class switching will only bring about the other subtypes, including IgG or the gamma isotype. Then comes antibody diversity. What is it generated by? So let's first look at the options. We have somatic hypermutation, allelic exclusion, protein splicing, interchromosomal recombination, or alternative splicing. Now, a lot of these options might seem very close. We know for sure that somatic hypermutation does contribute to diversity, but a few other options can also come very close. For example, alternate splicing might seem like that. What you need to keep in mind that antibody generation is very dependent upon the splicing of transcripts that occurs for those antibody or BCR genes. But diversity is not contributed by the splicing event. It is just to generate different isotypes of antibodies, not the diversity. When I say diversity, I mean with respect to the antigen to which that antibody can bind. That is brought about by those 7 events that we talked about, which include junctional flexibility, somatic hypermutation, etc. So in this case, the correct option would be option A, which is somatic hypermutation. Which one of the following is the correct sequential order of lambda light chain gene rearrangement of the antibody? So if you remember, we had talked about the light chain gene segment rearrangement and the heavy chain gene segment rearrangement separately because it occurs in the reason for that is because the gene segments that are present in each case are different. The heavy chain gene, it consists of also the diversity region, which is absent in light chains. So in case of light chain, we have VJ joining, of course, because D is lacking in that case, following which there is transcription, splicing, joining with the constant region, and finally translation. If you look at the other options, you can see some options have been switched about. Like for example, here obviously BJ joining occurs first. We know that for a fact transcription cannot occur first. So that eliminates option D. Next, if we look at the rest three options, we see that it is asking us whether VJ and C joining occurs first or transcription occurs first. So to eliminate any confusion in this case, you go back to the figures. It's always easier to remember what happens via the figures. So you see here that after V and J joining has occurred, there is the rearranged kappa or lambda light chain DNA, and there is immediate transcription. After transcription, we see that there is removal via splicing of the introducing sites and then joining of the BJ segment with the C segment and also with the leader peptide, the the black colored L fragment that you can see. So this sequence is also something you need to be very, very mindful of. You are currently learning, so obviously it's easier to get a little details or sometimes a little mixed up, but just keep in mind these figures. If you can, please draw them out so that it's easier to remember. So here, after BJ joining, transcription occurs, following which RNA splicing removes the interrupting segments, and that gives you the final mRNA, which undergoes translation or is converted into a polypeptide that finally forms the lambda light chain. So therefore, transcription is the early event, following which there is splicing. So that's where these options start differing. There is splicing, and then there is joining with the C segment, which is allowed to then undergo translation. So this would obviously be the correct option, option B. Then come the recombination signal sequences. If you remember, we had already defined them as RSS, and in the slides that I had uploaded, they were defined as, let me show you in the vectors. So here you found 2-turn RSS and 1-turn RSS, which consists of these different gene repeats or regions of repeats, which are heptamers and monomers, and they are present in different orientations, giving rise to different types of signal joint locations, or otherwise known as recombination signal sequences, which will generate different types of signal joint, which is what we saw here. We got a signal joint and we got a coding joint. In some cases, the signal joint was completely eliminated, which was in case of deletional joining, and in case of inversional joining, we have seen that it did not get completely excised, but there was a rearrangement of or inversional rearrangement that had occurred within the gene segment. So most of these questions have actually been covered. If not in the lectures, then definitely in the discussion session. So I that it was easy for you to address them. So here, obviously, the correct option is option A for heptamer and monomer. Then come the introduction of P-nucleotides during immunoglobulin gene rearrangement is an example of combinational diversity. Diversity caused by somatic hypermutation, junctional diversity, or combinatorial light chain heavy chain diversity. So obviously option D is not correct. We are not talking about combinations here, and similarly option A would also not be correct. You can address option B or C, but among them junctional diversity would obviously be the correct option, because if you remember, again, let me go back to the figure. Yeah. So here we have talked about junctional diversity, and we had shown that depending upon where the two RSS and the resultant BJ joining is occurring, based on where that joining site is eventually helping in the rearrangement, is going to then determine the specificity of the antibody. So for example, for this combination of gene sequences, they had shown all the possible combinations via which joining can occur. You can see that labeled as 1, 2, 3, 4, and 5. So each of these nucleotides can undergo recombination in such a manner that they can join with each of these 3, 4 nucleotides that are present here. The resultant rearrangements have also been shown here, but among them, not all of them are going to give rise to a functional polypeptide or a functional protein. Some of them will give rise to stop codons, which is going to be a sort of loss of function mutation, or you can say truncating mutation. Actually, sorry, that would be a truncating mutation, which is going to lead to the generation of a cryptic or a very short peptide, which will not be able to function. So these are non-productive rearrangements. On the other hand, if they are introducing diversity in frames such that the encoded amino acid is going to add something to the antibody, then that is a helpful rearrangement. Now, this junctional diversity or junctional flexibility includes the P and N nucleotide addition events, and therefore as the question has asked, that the introduction of P nucleotides is an example of junctional diversity. That the point at which there is a joining of different gene segments within the immunoglobulin gene, that diversity is coming about by the addition of P nucleotides. We have already come across how P and N nucleotide addition is taking place, so I'm not going to reiterate that. You can refer to the last week's lecture or even the discussion session for your reference. However, if there's a doubt, please let me know. We can certainly address it. Then comes the last question probably for the previous week. Yeah. So choose the incorrect statement regarding the 1223 rule. The first one is that the gene segment flanked by 12 base pair recombination signal sequence is joined to the one flanked by a 23 base pair RSS. Direct BJ joining to the immunoglobulin heavy chain gene segment is not possible. Direct B to J joining is in the TCR beta, TCR delta gene segment is possible, or direct joining is not possible. These are the different options that have been given. So BJ joining, we have extensively discussed the 1223 rule. It is otherwise also the one-turn, two-turn RSS rule that has been discussed in both the official lectures and in our discussion session. So for the 1223 rule, the incorrect statement has been asked here. So direct B to J joining in the TCR beta and TCR delta gene segment is not possible will be the correct option. So we have not, in the discussion session, we have not looked at the TCR gene segment rearrangement. But if you go back to the Sudeep sir's lecture, I think for the previous week, the last lecture had this part in extensive detail. So from there, you can refer it, or obviously you can refer to any textbook or any figure. You can just go to Google Images and study the 1223 base pair rule by a simple diagram, and it will clear this up for you. So these were the assignment questions that were given as part of week 3. If there are any doubts, please do let me know. I'll try to look them up. And apart from that, we have majorly been looking at BCRs or antibodies in the past week and past few weeks for that matter. For the last 2 weeks, this is what we have majorly been cited about. So because there have been different topics that have come in between, I thought this would be a good schematic to quickly recap what exactly happened. So if you remember, this is a schematic representation of this of the lymph node, which is the site of interaction of antibodies with the cognate antigen. So remember, the outer side is obviously the cortex, the inner side is the medulla, and somewhere in between we call the region as paracortex, as has been highlighted here. Now, each of the regions consist of different types of cells and are associated with very specific sort of functions. How so? First of all, let's look at the cortex. In the cortex, you see these green sort of granular structures, which are called as secondary follicles, or somewhere labeled as germinal centers, which are the sites where very active B cell division and proliferation is taking place. So that is what we see. B cell proliferation and differentiation is occurring in the germinal center. The smaller follicles, which is not secondary follicle, which is still less smaller in size, let's put it that way. The primary follicle is where B cell activation is taking place. Once it has been activated, it will undergo proliferation and differentiation and will be recognized as a secondary follicle within which germinal centers can be identified. Apart from that, within the paracortex, we see that initial B cell and T cell activation occurs, whereas in the medulla, plasma cell secretion of antibodies will occur, which will exit through the efferent lymphatic vessel and then will be circulated throughout an individual system. So this has been extensively discussed. If you remember, we had talked about how macrophages will present the antigens. We talked about the different centroblast formation, centrocyte formation, how there is affinity maturation which occurs, which leads to production of more efficient antibodies, which will be able to eliminate the pathogen much quicker. So this was the idea, just to sum up what we have been discussing over the past few weeks very quickly. Now, all of this is occurring. A pathogen has entered with first the innate immune response kicks in. It tries to eliminate the pathogen. It's not. If it is insufficient or incapable of doing so in a very efficient manner, then the adaptive immune response kicks in. So that involves humoral and cell-mediated immunity. As part of humoral immunity, the B cells, with the help of B helper cells, will produce antibodies. this is the overall timeline or the flowchart which takes place. If I'm speaking too fast or if there's any confusion, please let me know. I'll repeat it again. Please don't hesitate. So the different types of antibodies that are produced as a result of this have been shown here. You have already, I think I showed a similar image last week probably. So I think this was the image. This was the image I had shown to give you an idea about the overall difference in structure. But yeah, this is a schematic representation which simplifies the structure of an antibody. And this it's also a similar schematic only, but this actually shows you how the orientation of these antibodies actually work. So if you see, if you remember, they crisscross each other, which gives them their stability and which also helps in forming a very strong peptide binding cleft for um, easier recognition and easier binding to the antigen of interest. So if you see here, this is what I was actually talking about, that you will see the number of disulfide bonds will differ in each case. You will notice that each of them have consist of different domains of different colors, which means they have been encoded for by different gene segments. So this is IgG, then we have IgD, IgE, IgA, which forms a dimer, obviously, and there is a J chain or a peptide which associates between these two and helps in the formation of that dimer. And obviously the pentameric structure of IgM, which also has a J chain attached to it, which is a joining chain. And each of these monomers are actually linked to each other via disulfide bonds also, which forms a very large structure of the pentameric form of IgM. So these are the different schematic representations of the antibodies that we can have circulating in our system at any point of time in response to some pathogen, for example. Uh, Now, the question is that these are the different types of antibodies. Now, how can they actually circulate in our system? What brings about that change? So, as you might remember that BCRs or B cell receptors, which are present initially on a naive B cell, they are circulating in our system via the lymph nodes to look for an antigen of interest. Once that has been recognized and B cell activation or proliferation has actually been performed, the plasma cell, which is produced as a result, will produce secreted antibodies or secretory antibodies, which can be released into the system. How does that happen? So here you see the schematic which shows expression of secreted and membrane forms of the heavy chain by alternative RNA processing. Please keep in mind that this does not necessarily need to always be alternative RNA splicing. I have a better schematic to show the differences between these two. We will come to it in like 1 or 2 slides. But for now, you make sure that you describe this process as alternative RNA processing. What you see in the figure on the left is the amino acid sequence of the carboxy terminal end of the secreted and membrane heavy chains. We are only looking at the heavy chains here because that is what determines the isotype of an antibody, which is what we are concerned with right now. The residues have been indicated by single-letter amino acid codes. Hydrophilic and hydrophobic residues and regions are indicated by purple and orange respectively. Charged amino acids have a plus or minus, so that actually has got omitted. But yeah, it's indicated by plus or minus. So among these hydrophilic residues, which are water-loving residues, they are, which means that they are soluble in nature and they can remain in contact with water or basically can remain exposed around the surface because the physiological condition is aqueous. So any amino acid or any protein for that matter, which is circulating freely in the system, will always have hydrophilic residues on the outside. Hydrophilic means water-loving residues on the outside, and or polar residues on the outside, and hydrophobic or water-scared or water repelling residues on the inside, or will be put towards the core of the protein. So suppose you have a polypeptide which is synthesized like this. When it folds, it will form a structure such that what is exposed on the outside will be hydrophilic and what you ensnared in within the core, that will be hydrophobic in nature. Just a second. It's fine. So yeah, that's how the structure actually works. Now, if you look at these representations, what you see on the left is the secreted form, and what you see on the right is the membrane-bound form of IgM. It has been labeled as membrane mu and secreted mu. So for the secreted mu, you will see that most of the residues that you see here, they are hydrophilic in nature, as has been shown by that purple color, and the orange residues are hydrophobic. So this is actually an expanded version of the peptide which has been shown here, and this is encoded by the S exon of C mu. Where is the S exon? It's actually a part of the secreted exon, which is shown here for the secreted antibody. We'll come to it, don't worry. So what you need to keep in mind is that these are majorly rich in hydrophilic residues. What you see in between are engulfed towards the center when they eventually fold. On the other hand, if you look at the membrane-bound antibody composition, you will see that most of it, first of all, it's very long, and most of it is very rich in all of these amino acids which are hydrophobic in nature, and that is necessary if it needs to be bound to a membrane. Why so? If you've studied, I hope you're familiar with it. If you've studied the structure or the composition of the plasma membrane, you know that would know that it is made up of phospholipids and forms a bilayer, and these phospholipids are amphipathic in nature, which means that they have both hydrophobic and hydrophilic entities. So just to if you have already studied the structure of these phospholipids, you would know that they consist of a hydrophilic head and two hydrophobic tails, and one of them usually has a kink based on whether or not it is a saturated or an unsaturated amino acid. I think it's too far gone, not amino acid, lipid molecule. So based on how this is arranged, you get a layer where the outside, so this is a phospholipid bilayer, the outsides are always hydrophilic in nature, the center part of it is hydrophobic. So proteins that need to be embedded within the membrane, they don't have better representation. Say, for example, this is the plasma membrane. So the outer sides of it are hydrophilic, and this thickness that you see in between, that is hydrophobic. So any protein that needs to be embedded within the membrane, that needs to have a region which is hydrophobic so that it can be stably embedded within the membrane. What is exposed on the outside, which is in the extracellular side, can be hydrophilic. Anything that is inside the cell can also be hydrophilic in nature. So that's how you see the arrangement also, that this is the transmembrane and these are the extracellular and cytoplasmic regions of the membrane embedded IgM. The question now comes that how can this and all be brought about? If you again come here, this is the gene segment arrangement of the primary heavy chain transcript, then you would see first of all that VDJ recombination has already occurred. Once this has occurred, what you now need to look at is the recombination that will take place with different types of constant regions that will determine the isotype of the antibody. There we see that there is an extensive region of C mu and an extensive region of C delta. The rest of it has not been shown. We have zoomed in onto the C mu and C delta regions. Within C mu, we see several subregions, C mu 1, mu 2, mu 3, mu 4. A dark blue segment, which is known as the S exon, which encodes for this, what you just saw, the secreted mu exon. And it is followed by M1 and M2, which are transmembrane region. You see here that both the S exon and the M2 exon are followed by very small black colored fragments, which are known as sites of polyadenylation. So I'm guessing that you would already have some context about the processes that are occurring as part of the molecular biology of any cell. So after DNA is transcribed, a number of post-transcriptional modifications are extremely necessary to give rise to the mature transcript, and that mature transcript is only allowed to undergo the process of translation. So polyadenylation and splicing are two crucial events which actually regulate this. So in case of secreted versus membrane-bound antibody, it is not just splicing which is necessary, but rather it is the process of polyadenylation which is extremely crucial. If polyadenylation occurs within site 1, which is polyadenylation site 1, then what do we see? We see that RNA transcript or secreted mu region, because the transcript will get terminated right here. As a result, M1 and M2 regions will not be transcribed, and what you get as a resulting mRNA because of RNA splicing is the one that encodes for the mu chain, which is what we saw here. On the other hand, if polyadenylation occurs at site 2, then that will give rise to a polypeptide upon splicing, which consists of M1 and M2, which are these transmembrane regions, and that will allow the antibodies to stay embedded. This is only the example for C mu. C delta also undergoes a similar process, and once IgM and IgD have been encoded, there can be isotype switching, which can give rise to all other types or subtypes of antibodies. So coming back to the legend here, the structure of the primary transcript of a rearranged heavy chain gene, which shows the C exon and the polyadenylation sites. Polyadenylation at site 1 or 2 and the subsequent splicing will generate mRNAs encoding either secreted or membrane-bound chains. So I hope this concept is clear. It's what you need to keep in mind is the sequence of events which are occurring. That first, from the hematopoietic stem cell, what are the events that have occurred? I will not go into details about it now, but I'll just show you the figure which will recap it quickly. Yeah, so this is the figure I want you to keep in mind, that what are the events that start from the hematopoietic stem cell, lymphoid, pro-B, pre-B, mature B cell, and at the mature B cell, we only have the MIGM expression. From there, the mature B cell, upon antigenic stimulation, will form an activated B cell, which will either form IgM secreting plasma cells. When you have SIGM, that means these cells have antibodies with the S exon. Upon polyadenylation is occurring after the S exon, so all of their antibodies are getting secreted, whereas some would be kept behind as memory B cells. Apart from that, they can undergo isotype switch to form different types of antibodies. So this is just to comprehensively correlate all of the information that we have been studying till now. It's very important to do that because you're getting a lot of information, and what you need to keep in mind is that these are simultaneous discoveries done by huge groups from across the world that have been taking immunology as their main target research for the past, I don't know how many years, but several decades. So as a result, information is very scattered. What you need to understand is how to organize it and how to make sense of it. So obviously reading books is the best way to do that. But yeah, you can obviously refer to any paper also if you want to just be updated. But keep in mind that your basics should be absolutely clear. So this is the logic behind the generation of secreted and membrane-bound antibodies. Now we come to the next concept, the role of post-translational modifications in immunology. Why I specifically got into it is because we are talking about polyadenylation and talking about splicing extensively. So polyadenylation, I hope you already know that it is the process where at the end of any mRNA, not all RNAs, but mRNA specifically, which have to undergo translation, which are from coding genes, a stretch of A nucleotides or adenylation basically occurs, and a stretch of A nucleotides are added, and that is exactly how it is recognized as a mature transcript and how it is then considered to be taken forward for the process of protein generation, and then the ribosomal assembly takes place and so on and so forth. But when we talk about splicing, why I specifically said in the previous slide here that you have talked about alternative RNA processing and not alternative RNA splicing is because that alternative splicing events are of many different types. This schematic on the left is exactly what represents it. Let's go over it one by one. They are very similar, but not exactly the same. The first one you see here represents constitutive splicing, which is where all of the introns are, which is the textbook definition of a splicing event, that it is the process where introns are removed and exons are joined with each other to give rise to the coding sequence or the exome of any gene or of any individual or any cell. So that is giving rise to the exome that you see on the right. Second case is where there are mutually exclusive exons. This is actually something that occurs very frequently in immunology and also in other developmental processes. So this can be a good task for you to actually look at. Look at the concept of alternative or mutually exclusive splicing in other developmental processes. I'll give you something for reference. If you have studied the process of sex determination in Drosophila, then it's a fantastic concept and it's very it's very subtle and very beautifully studied as well. So that is so heavily dependent upon the process of alternative splicing that it should blow your mind. You can try looking into it. That it's just the way in which transcripts are being regulated that is determining whether the Drosophila will become a male or a female. So it's not the same, obviously, for not exactly the same for humans, but yeah, just as a process, it's fantastic, and I would really urge you to go ahead and read it. So yeah, coming back here, what you see in figure B is a mutually exclusive splicing event. So that means there are two possibilities. If this is the gene segment that is present for a particular gene, or this is the arrangement of the gene for any particular location, then in that case, any of these two events can occur, and you can actually label them as event one or event two. If you remember the way in which junctional diversity was described, there were five possibilities that were taken into consideration. So based on that, this can be considered as event one, and this can be considered as event two. So if event one occurs, then this joining, the first exon and the third exon, along with the fourth exon, will be forming the final transcript. And if event 2 occurs, then the first exon joins to the second exon, and the second joins with the fourth. So these two exons, exon 2 and exon 3, are mutually exclusive. It means that under normal circumstances, this particular gene will either contain exon 2 or exon 3, and therefore they are mutually exclusive in nature, and that you can see in the genes. So this is a very crucial event. It means that it has some very significant role to play, which is why this exon skipping occurs. How can this be executed? That's a different event. I will explain what is the possible way in which this regulation occurs, but yeah, it can be very gene specific, very gene specific in some cases. I'll explain that very soon once we come to these splices of assembly, just for reference. The third type is called as cassette alternative intron. So it's very similar to the mutually exclusive exon event, but here it's actually cassettes which are taken into consideration, that it can either be these two which are getting joined, or including this second cassette also, it can be joined. Then come the event where it is alternative 3' splice site, which means that within the same exon, there are two different sites at which splicing can occur. There is a site which is lying within that particular exon, which which can also be used as a splicing site. As a result, if there is normal splicing, regular splicing, then it will lead to this particular condition where you have these two gray colored exons and the white exon in between. However, if the alternative splice site comes in between, then only this particular region and the middle part will be excluded, and this will be the final transcript that you get. So this is an alternative 3' splice site. This also occurs for several genes. Again, if this is interesting to you, you can go ahead and look at examples. There is extensive gene regulation which occurs in all sorts of living organisms. Why only humans for that matter? Similarly, alternative 5' splice sites can also exist, and that gives a similar result. And finally, there can be some cases, very, very rare cases, where there is intron retention, which means that the black colored intronic region that you see here is not getting removed, possibly because it has some regulatory role to play in these individuals or for that particular gene, and therefore it is retained in the final transcript. So these are the different types of alternative splicing events that can occur. The question is, why do they occur and how exactly do they occur? So that for that, I think you would need to have some background about the way in which splicing occurs. So what you see as representation is the assembly of the spliceosome. Spliceosome is a machinery which consists of several different types of proteins and RNA for that matter. RNA and protein together form something known as RNP, which is called as ribonucleoprotein. So what you see here as RNP, they are all ribonucleoproteins, which are machineries which can work as enzymes. And they are responsible for this cutting of DNA and the removal and the joining, all of these events. So this is a schematic representation of the sequence elements and the proteins that are present upon an RNA transcript at the exon-intron boundary. So it is actually the assembly of the spliceosome which delineates. When I say delineate, it means it defines that this region is an exon and this is an intron. So if you see in all of these cases, you are seeing these events where splicing is occurring. The point is, how does the system know that this is where splicing needs to occur? that definition or that idea is explained by spliceosome assembly. So if you see here, these are exon intron and exon intron junctions. The assembly of the spliceosome at these two places easily defines that what is present in between. That region is the intron and that needs to be excised. If there is no assembly at one particular exon intron junction and then there is assembly at the next one, so then that exon along with the surrounding introns will be retained and splicing will be occurring at the next event. So it's simply where the spliceosome assembles. Based on that, splicing will take place. Apart from that, this assembly is not something that happens at will. It is controlled by several cis and trans elements. Cis elements, it means these are cis sequences. They are DNA sequences. Trans elements mean that these are something that are coming from somewhere external. They are some protein or some RNA which is coming externally and regulating this process. So there are some factors which are called as trans factors, and there are elements which are known as cis elements. Uh, the coordination between these two is actually what regulates several types of gene expression events, not just splicing, and it is an example of what we see here. The enhancer elements, known as ESEs or exonic splicing enhancers and intronic splicing enhancers, are recognized by activator proteins and silencer elements. Intronic splicing silencers are bound by repressor proteins. So basically, you are promoting, when it is a region that needs to be spliced out, you're promoting the process of splicing and you're repressing the region which silences splicing. So there is simultaneous regulation of both, so that splicing can be promoted and therefore that region gets excised. So these two protein families engage to promote or inhibit spliceosome assembly at weak splice sites respectively. So based on how this assembly is being regulated, based on that, a particular region will either be carried forward in the final transcript or will not be. Why I went into this much detail is because this event of splicing plays a very, very key role. Like we have extensively been seeing all of these events that we are so simply showing by just a schematic that, a triangle is showing that this region is getting excised. It's not as simple as it seems. There is a lot of regulation, a lot of ATP that is invested, a lot of checkpoints that have been put into place to ensure that the immune system works and works perfectly. So with this in mind, I just wanted to give you some insight into the process. I hope it was somewhat understandable. If it is an advanced concept, please let me know. I am going forward with the assumption that you are familiar with splicing. But yeah, sometimes we do not know these extra details about splicing and the variations that occur within it. Mostly we are familiar with alternative splicing, but all these different subtypes are also important to know, especially if you are thinking of going forward in this field. So next we'll come to the subtypes of antibodies. Now we have been seeing isotype switching for a while now, because it is a process that is helping in the transition from one type of antibody into another. In context of immunology, an isotype would be a category of antibodies that is defined by the heavy chain constant region or the HC region and other physicochemical properties such as molecular weight. Each isotype contains a unique heavy chain constant region, and there are 5 antibody isotypes which we have obviously talked about: IgG, A, M, E, and D. IgG is the most common isotype. So obviously the definitive region, what you see in the leftmost diagram in each of these cases shows the region which gives the distinction between each of those definitions. So when the difference lies in this region, which is the heavy chain region, that is giving rise to different isotypes. Examples being IgG1 and IgGM from mouse. Okay. And obviously, as a result, what you see, the surface epitopes in each of these cases is also different. The second definition comes for allotypes. What are allotypes? They are antigenic determinants, which are specified by the allelic forms of IgG genes. IG genes or immunoglobulin genes. Why is that relevant? It's relevant because we as humans, or most animals for that matter, are diploid in nature. As a result, we carry 2 copies of each gene within our body. As a result, we have 2 types of gene segment rearrangements which are occurring, and therefore for each condition or each subtype, we have some differences, very subtle differences, but some differences which are present in each case. In context of immunology, Allotype is an immunoglobulin or antibody variation, which is in addition to isotypic variation that can be found among an antibody class, not in interclass, but instead, for example, if we talk about IgG, as has been shown in the schematic, within IgG, there are several subgroups. Among that, IgG1, if we take into consideration, there is strain A and strain B. So there will be differences between those two. Of course, there will always be difference between a mouse IgG1 and a human IgG1 also, for example. They will not be the same. So there is a difference that exists between the surface epitopes of these antibodies in different conditions. So this can be found among antibody classes and is manifested by the heterogeneity that exists for immunoglobulins present in vertebrate species. In fact, when we talk about for that matter, sometimes blood transfusions can also be taken into consideration when you're taking blood from another individual. And there are some, for example, if in case you get some there is antibody which is crossing in the case, or sometimes during deliveries there are cases where this occurs, where there's a reaction to the introduction of another type of antibody from another individual, or in fact, the best example would be in case of a transplant. Then allotype matching is very, very crucial. And it can especially be a very huge problem if it is extremely similar, if the antibody that has gotten introduced into a person is very similar to their own antibody. What happens then is that while that external antibody is being recognized as foreign, the immune response to it, while mostly specific, can sometimes go wrong. Say, for example, I am, for whatever reason, my body is facing another antibody, which is 90% similar to an antibody that I have in my system. Say, for example, IgG1 only. So I have some IgG1 to some pathogen, for example, some bacterial infection. I have it circulating in my system already. In case there is some antibody which is very, very similar to the antibody that I already have, then it can elicit a very wrong immune response and it can remove something from my system. Even worse, if it so happens that the antibody has some epitopes which are very similar to some cell surface receptor of my own cells, then that can also be a problem. So allotype matching needs to be done to prevent any incorrect or very abrupt immune responses to any foreign entities. The third example is of idiotype. So as you can see by obviously the first diagram, that the idiotype mainly looks at the topmost area, which is the variable region of the antibody. And the surface differences or surface epitopes that are present over there will will be for different types of antigens. So what you see here, the mouse IgG1 against antigen A and mouse IgG1 against antigen B will have different idiotopes, which is a epitope of different idiotypes of antibodies. So they are antigenic determinants of immunoglobulin molecules that are located at the variable region of antibodies. So at the topmost region. So when at the topmost region, they are called as idiotypes. When in the constant region, they would be encoding for isotypes. When they are for different conditions from different alleles from different species, they would be allotypes. So for idiotypes, we are saying that they are further subdivided into those that reside at the antigen binding site, which is the paratope of the antibody molecule, and those which are at areas adjacent to this site, the framework determinant. So that's not very relevant to you. For now, we can just focus on the differences and the definitions of isotype, allotype, and idiotype. So I hope it is clear. It was already explained very well by Professor Sudeep in his lecture only, but I just wanted to iterate it with a different schematic if it helps. If any doubt is there, please let me know. I know this can be a slightly confusing concept, especially allotype. So if there's any doubt, please let me know. I'll look for even better schematics to explain it. So yeah, again, you can interrupt me at any time. I'm going ahead, but if there's a doubt, please interrupt. So with that in mind, now we move forward with the different antibody subtypes. In particular, if I am to be more technically accurate, different antibody isotypes. So we just talked about the definition of isotypes, and you saw that these are all of the different subtypes that exist. Among them, you see that maximum variation can be seen for IgG, which has IgG 1, 2, 3, and 4. Then you have IgA, which has IgA1 and IgA2, IgM, E, and D. They have only one subtype. So among them, you can see in each case, what is the heavy chain component that is giving rise to the antibody? What are their molecular weights? You can see that IgA1 and A2 can have very variable molecular weight. So what is the reason for it? If you just look at the legend at the bottom, then that actually will show you that molecular weight in each case can be quite variable. IgG, E, and D always exist as monomers. IgA surprisingly can exist as a monomer, dimer, trimer, or tetramer. We here have only talked about the fact that IgA can exist as a dimer. If we just quickly go back to this schematic, yeah. So this is the dimeric form that has been represented, and for IgM we had looked at the pentameric form. However, IgA has the capacity to exist as mono, di, tri, or tetramer. Membrane-bound IgM is a monomer, but secreted IgM in serum is a pentamer. So based on that, its molecular weight will also vary. If you look at here, it appears to us that IgM molecular weight has been shown for the pentamer. So if you just want to get the single molecular weight, you divide it by 5 and that should be good enough. You see the normal serum levels, and you can see that it's quite variable in different isotypes, that the level of antibodies that are present in the serum is very different. If you carefully look at them, the lowest level can be seen for IgE. And I think we had discussed why that is important. It's because the IgE is responsible for triggering the inflammatory response or the hypersensitivity immune response. And therefore, its basal level within the blood or within an individual at any point of time should be under extreme control in order to avoid very sudden hyperactivation of immune response. So unless you have an allergy, unless you have been exposed to some sort of antigen which is triggering an immune response, your IgE level should be very, very low. Similarly, IgD, IgA2, IgG4, all of these should also be low. The rest of them seem to be quite significantly abundant within the system. Then you have the serum half-life, which means how long do they actually circulate within the system before they are cleared from the system. Then comes the category of whether they're able to activate the classical complement pathway. We will get into details about the complement when we come across it in the course. It's not something that is done right now. And you can see that not every antibody subtype is capable of triggering the complement response. Only certain specific subtypes are. Majorly, you see IgM and some subtypes of IgG as well. What you also need to keep in mind is that the number of plus or minus symbols that have been added also show you the intensity of the response or the activity of that response. So IgM is the main player that mediates the classical complement pathway, as you can see from this tabulated form. I want you to keep in mind because we will be taking up a complement system also in a few days, so it will be relevant then. Whether or not it can cross placenta, you see that all of the IgG subtypes are capable of crossing the placenta. None of the other subtypes are able to do so. Then comes the criteria of whether or not it is present on the membrane of mature B cells. So for mature B cells, we have IgD and IgM discussed already. Whether they bind to Fc receptors or phagocytes, then majorly it's IgG, not the other ones, but for IgM, it's questionable. Mucosal transport, this is not very relevant to us right now, but yeah, mucosal transport can be triggered by IgA. So we'll come to it later, why mucosal transport is necessary. And finally, it uses mast cell degranulation. This process is very crucial for the hypersensitivity response, and therefore IgE is the only subtype which is capable of doing that. And that mast cell degranulation is a precursor step towards the release of histamines and then triggering an entire elaborate immune response against it. So yeah, and apart from that, one extra information that you can see in the legend is that IgM is the first isotype that is produced by a neonate, and also the first isotype that is produced during a primary immune response. So it is the default transcription pathway that is present in a normally developed individual. So yeah, this is a comparison that shows this. What you see on the right is a representation of the different IgG subtypes. So while they majorly look similar, it's the middle part that shows a lot of difference among these subtypes. So the number of disulfide bonds and the location of the disulfide bonds are different in each case, which gives rise to IgG 1, 2, 3, and 4. So IgG3 has the maximum number of disulfide bonds. That's exactly why I told you to keep a check on what is the number of disulfide bonds that are present when we were discussing previous week's questions, assignment questions. So obviously here you can see that the maximum disulfide bonds you can see are for IgG3. It's, I think, even more than what you see in the pentamer. I'll have to check it once. I think the IgM pentamer has more disulfide bonds. I'll just check it once. But this by itself within a monomer, it has the maximum number of disulfide bonds. So yeah, this was a comparative just recap of all of the different antibody isotypes. Now, very briefly, how does the process of isotype switching occur? If there is any doubt again at any point, please interrupt and please ask. We need to have an interactive session. I can already see very few people are joining these sessions. I understand that you have access to the lectures and that's obviously very helpful. But yeah, coming live and actually interacting, coming up with questions, it's always more beneficial because I otherwise don't get to know what your doubts are. So I'm just coming up with information that I think is relevant, that I think needs to be clarified. But if there are doubts, please ask. So
Excuse me, ma'am.
yeah.
Ma'am, I think every antibody crosses the placenta, right? Ma'am?
Uh, Not always actually. There are so I think this was discussed in the lecture by Professor Sudeep also. Is there some confusion because of that?
Yes. No, ma'am. I
Okay,
think every antibody crosses the placenta, but here most of the antibodies are
right.
minimally
So
put.
okay, yeah. So from what I could hear, I think your question was about whether you somewhere. I think you might have read it somewhere maybe, or you're speculating that every antibody crosses the placenta.
In the lecture, ma'am, in the lecture.
Okay, so I'll verify it once again. I also think it might have been there in Professor Sudeep's lecture. So this is again an older edition of Coupe. But from what I had read initially, that IgG is the subtype that has maximal crossing of the placenta. I'll double check it. It could be that this is an error on my part. But from what we had done, what we had studied in our lectures also, was that transplacental migration is possible only by IgG, and that is controlled by the fact that IgG is most abundant because of the way in which IgG is arranged. But since you're asking, I'll double check it. I will not comment on it since it might have been an update and it's my mistake. I should have double checked it. But yeah, I'll look into it and get back to you on it. But based on Kubi, this is the
Okay.
information that was given. So yeah, you pointed out a good fact. Let's just go back to Kubi right now and see. So I have it open. Just it'll just take a minute. So here, as again, this is an older edition. I'm not claiming anything. I will double check it. But here's what has been told that the level of naturally acquired maternal IgG, which the fetus acquires by transplacental transfer, remains high for about 6 months after birth, and that is exactly the time where immunization or vaccination is carried out to shoot up the level of antibody that that person or that the child will have for an extended period of time, and then they will have memory cells in response to that for a very long time. But yeah, I'll still check it. Yeah, I'll check it. That's all the information that is given here. I'll check it and get back to you. Thank you for pointing it out.
Okay,
Coming
ma'am.
back here. So we were talking about isotype switching. So how is it exactly occurring? We talked about the fact, I think this might, no, okay, we did not discuss this previously. We just talked about the fact that isotype switching occurs. The point is, how does it occur? This again has been taken up by Professor Sudeep during his lectures, but basically it's again the process of rearrangement which is occurring. Majorly, he talked about the fact that cytokines regulate this process, and in particular, here in the book, the only cytokine that has been mentioned here is IL-4 or interleukin-4, which induces class switching in a very successive manner. Let's go over the legend first. So first, it induces. First, it goes from C mu to C gamma 1, and then from C gamma 1 to C epsilon. So this is the arrangement of the heavy chain region, and from there class switching needs to occur, and that switching is taken care of by the switch regions that you see that are present in between the different constant regions. So there is S mu switch region for mu, which includes both mu and delta. Then switch regions for gamma 3, gamma 1, gamma 2b, and gamma 2a. Also for epsilon and for alpha. First recombination occurs in, or first rearrangement occurs in a way, or looping occurs in a way that involves C mu and C gamma 1, and then it goes from C gamma 1 to C epsilon. So there's a successive removal of DNA loops and recombination that occurs, and that allows us to control the switch without making it a very abrupt process. It could have also occurred in a way that S mu and S epsilon undergo that switch directly, but that A would have been more error prone, would have been energy intensive, and a very big DNA loop is very difficult to stabilize without the involvement of a large number of proteins. So this is a sequential step which is taken care of, and it can be regulated at each step as well. So examination of the DNA excision products which are produced during the process of class switching from C mu to C gamma 1 showed that a circular excision product containing C mu together with the 5' end of the gamma switch region, which is S gamma 1, and the 3' end of the mu switch region, which is S mu, was generated. So that actually has S gamma 1, and it has C mu, which are it has the two ends. So how do you identify how the switch has occurred? It is basically that people started studying the excision products which are generated as a result of this looping, and they looked at what are the regions that are produced as a result. So when this DNA loop was produced, this is the excision product that they got. And in this excision product, they found this short blue and pink colored segment that you can see here, and that contains switch regions from both S gamma 1 and S mu. So here is S mu, here is S gamma 1. There is a joining of these two regions, so they found a recombined site. Furthermore, the switch from C gamma 1 to C epsilon, which means from IgG1 to IgE when the final switch is occurring, this produced a circular excision product that contains C gamma 1 together with portions of the other switch regions. So when the second looping and recombination event takes place, then what they got as an excision product, resultant excision product is consisting of epsilon and gamma 1, and that's how they decoded. So this is just to give you an idea of how experimentally you can figure out what exactly is happening, because people must have studied when whichever scientists were working on this, they might have observed that yes, there's a switch from IgM to IgE. The question is, how does it happen? You substantially and very sequentially you study with experiments what are the products that you get as a result of the recombination, and you decode or you trace it back to what might have been the event that has occurred. So this is what was studied, and this is how, based on the sequence of genes or the gene segments that are present, the entire process was decoded. So class switching effectively depends upon the interplay of three elements. You have switch regions, you have a switch recombinase, and the cytokine signals that detect the isotype to which B cell will switch. So, okay, this was the event of isotype switching. The rest of the details have been explained by Professor Ghosh. So I did not see the idea of repeating the whole thing. I just wanted to give you an idea that experimentally, how can this be done? Again, if you want to read how the experiment was performed, you can go to Kubi, you can go to the original paper, and that will give you some insight into how this can be decoded. Next comes the second topic that was taken up as part of this week, which is the major histocompatibility complex, or MHC. So what is the need for the MHC? MHCs are actually a collection of genes that are present within a stretch of DNA. They are present one after the other, and they are kind of present in tandem, and their location is at chromosome 6 in humans and on chromosome 17 in mice. MHC is referred to as HLA or human leukocyte antigen complex in humans and the H2 complex in mice. The arrangement is somewhat different, but their organization is effectively the same. Now we will only be for today, we'll be only discussing MHC1 and MHC2. 3 has not been taken up in the lecture, so I don't want to unnecessarily introduce it. But I just want you to know that there are 3 types of MHCs and they are arranged in this manner. So you don't need to remember all of this detail. It's just for context. You can see that MHC class 1, 2, and 3 are present in mouse, and MHC class 1, 2, and 3 are present in humans. Their regions are different. All of this detail you don't need. What it effectively produces as gene products is what you need to see: HLA, A, B, C, complement proteins, TNF alpha, beta, and these are TPDQTR. Too much detail, you don't need to know it right now. It's just to show you the arrangement, how it exactly is present in the genome. So class 1 MHC genes, what do they encode for? They encode for glycoproteins that are expressed on the surface of nearly all nucleated cells. The major function of class 1 gene products is presenting the peptide antigens to DC or cytotoxic T cells. Class 2 MHC genes encode for glycoproteins that are present primarily on antigen presenting cells. Keep in mind that MHC class 2, it encodes for glycoproteins. Both of them are encoding for glycoproteins, but particularly class 2 MHC is only present on antigen presenting cells, in particular known as the professional antigen presenting cells. So this difference was also highlighted by in the uploaded video lectures. So macrophages, dendritic cells, and B cells are the professional antigen presenting cells. There was a comparison that was shown in their abilities. I think I forgot to include that table. Maybe I'll include it in the slide when I upload it. In Kubi, you'll find a comparison between these between the efficiency of these antigen presenting cells in doing their job, how B cells, macrophages, and dendritic cells are efficient. Among them, obviously, dendritic cells are the most efficient at this job. So they present their antigenic peptides to Th cells. And finally, class 3 MHC genes, which we are not getting into today, they encode for, in addition to other products, they encode for various secreted proteins that have immune functions, including complement activation, molecules involved in inflammation, etc. TNF alpha and TNF beta are also included as part of that. We are not getting into right now, but yeah, these are the 3 classes of MHC genes that are present. For now, let's keep this in mind and let's move ahead and look into the structure of MHCs. So obviously you're already familiar with it, but this is just another representation of the same concept. So if you look at class 1 MHC, you will see that it has one long polypeptide chain or protein. And along with that, there is another non-covalently attached protein molecule, which is known as beta-2 microglobulin. So this is a schematic diagram of class 1 and class 2 MHC molecules showing the external domains, the transmembrane segment, and the cytoplasmic tail. And we can also see where the peptide binding cleft is present. So that is also extremely important in this case. In case of class 1 MHC molecules, you see that they have alpha 1, 2, and 3, which are part of the same polypeptide chain attached to a transmembrane segment and a cytoplasmic tail. So there is one polypeptide which is kind of bending like this and creating a cleft for some antigen to come in, some peptide to be bound, and for that to be presented on the surface. To give it structural support and to ensure the integrity of MHC class 1, there is another beta-2 microglobulin which is present very close. On the other hand, for class 2 MHC molecules, this is the easiest way to remember. Class 1 only has one continuous peptide contributing to the peptide binding cleft. Class 2 MHC molecule has 2 peptides that are present in parallel. So one contributes alpha 1, alpha 2, another contributes beta 1 and beta 2. Both of them have intra-chain disulfide linkages, not interchain, intra-chain disulfide linkages, which you can see here extensively. That stabilizes the structure, both in alpha 2, alpha 3, and beta 2 hydroglobulin for MHC class 1. And here also you can see the same. And the most important, most distinguishing factor between these two is the peptide binding cleft, which is present here between alpha 1 and alpha 2 from the same chain. And here it is present between alpha 1 and beta 1 from two different chains for class 2 MHC molecules. Now, how does this come about? Just a second. How does this come about? How are we getting all of this information? How can we eventually get those polypeptides which are encoding for the final MNCs? So again, coming back to the gene arrangement that is present in each case. For MHC1, you have obviously got a leader peptide because that will direct the protein. You have alpha 1, alpha 2, spaced out by a long stretch with alpha 3. You have a transmembrane region and two constant regions. All of the intronic regions are removed. What you get as a result in the final mRNA is L alpha 1, alpha 2, alpha 3, transmembrane, and two constant regions followed by polyadnylation. Each of those parts is contributing to the final structure. You can obviously correlate the two. The two constant regions are present, not the constant, sorry, these are cytoplasmic regions. The two cytoplasmic regions are present on the inside. The transmembrane region is present in that phospholipid bilayer, which is what I was trying to explain some time ago as well. And then you have the alpha 3 interrupted by a long stretch, not a long stretch, but interrupted by a region which is not into an immunoglobulin fold or any fold for that matter, followed by the alpha 2 region, which has a huge disulfide linkage. Then comes the alpha 1 region, and that ends the final protein. This generates the peptide binding cleft. Attached to it is beta 2 microglobulin, and that also contains the another disulfide linkage. And there is correspondence between the exons and the domains in the gene products, which is what we have tried to show here. On the other hand, for class 2 MHC molecule, you see obviously that there's a difference in arrangement. You will see that it contains two different polypeptide chains, one which is shown at the top, one which is shown at the bottom. So at the top, you are seeing the beta chain. So that has beta 1, beta 2. What you see here is one exon which contains both transmembrane and cytoplasmic regions within the same entity. So it is shown as TM plus C. So it has some region which is present within the membrane, and within attached to it, you have a cytoplasmic region also. Along with that, you have two cytoplasmic regions attached as well. So this is also another distinguishing feature. So if you're ever listing it up, this is how you can try to do that. And with that, in combinatorial association, you have the alpha chain as well. So that has alpha 1, alpha 2, TM plus C, and only one constant region. So these can be some things you might have to keep in mind. The number of constant cytoplasmic regions, sorry, I keep saying constant, cytoplasmic regions, the way in which the transmembrane region is arranged, is it exclusively transmembrane, or does it also have some cytoplasmic side? That's also something you can keep in mind. And then they both associate with each other to give rise to the final class 2 MHC molecule. Keep in mind that beta 2 microglobulin is not being encoded by the same gene segment or anywhere close by. It is encoded, or it is transcribed and translated separately. It is located even on a different chromosome, and it just forms in association to give rise to the final MHC1 molecule. Now, the subtle differences between peptides, the efficiency of peptides and their binding by MHCs. So this obviously shows a comparison between the ability of peptide binding between the two MHCs. So class 1 molecules they have the peptide binding cleft formed by alpha 1 and alpha 2. And this is called a sort of a 3D representation, a space-filling 3D representation of the way in which the proteins or these MHCs actually bind to their peptide. We'll come to the nature of it. So this is for class 1 MHC. And for class 2, you have alpha 1 and beta 1. The peptide binding cleft nature is also different in each case, such that in case of class 1 molecules, it is closed at both ends, whereas for class 2, it is open at both ends. What does it mean? It means that the way in which the peptide is bound, suppose this is a surface, this is the peptide binding cleft. On that, the protein is bound like this, or the polypeptide is bound like this. it's closed or it is anchored at both ends when it comes to class 1 MHC. For class 2 MHC, it's sort of bound like this. I think I would like to, I don't have it in the presentation, but I would like to show that. Um, so just give me just a minute. Yeah, so these are different representations. Yes, this is the diagram I was looking for. So this is the conformation of the peptides that are bound to class 1 MHC molecules. Let me zoom in a little bit more. So this is this shows you the conformational difference in peptides that are of different lengths. So when there are longer peptides, they bulge in the middle. Shorter peptides are more extended. Contact with the MHC molecule is by hydrogen bonds to some anchor residues. We'll come to those anchor residues soon. So this is how the protein eventually binds. If it is a shorter peptide, it will be quite closely associated. A longer peptide will keep on bulging, but it will be anchored and therefore it is closed at both ends. Um, why is this so relevant? It's important because if you think of it in a three-dimensional manner, if a protein is more, or if not protein, if a peptide is more attached to the MHC versus if it is bulging out, the way in which it will be presented to the cell that will recognize that peptide will also be different. So its affinity or its tendency, if we look at it in a structural manner, in a 3D conformational manner, that will also be different. So therefore, so much stress or so much research has gone into understanding the way in which peptides are presented to the cells that will execute the immune response to those antigens. So this is a schematic representation. What you see here are molecular models that are based on a crystal structure of an influenza virus and antigenic peptide and an endogenous peptide bound to class 1 MHC molecule. And this is also a similar representation of how exactly they bind. They form this sort of bulged out sort of structure. And MHC also they had. Yeah, so this is actually okay, we'll avoid it for now. It will be too advanced. Let's come back and wrap up this topic. So the way in which class 1 MHC peptides the way in which class 1 MHC binds peptides is in sort of a closed manner, like I just explained, versus if you look at the class 2 MHC molecules, then they are more open, there is more accessibility, and it is able to bind to longer peptides. How much longer? Approximately 13 to 18 amino acids, versus for class 1 MHCs, 8 to 10, or like Sir had mentioned, 9 to 11 amino acids are usually the length of the peptides that can be encompassed by class 1 MHC. We also explained the reason behind it, that if we look at the MHC structure, if it is coming from the same same if they're coming from the same chain, which is the case of class 1 MHC, then the cleft or the dip that can be generated in that case will only be limited. Versus if they are coming from two different entities. He explained that with the help of like our hand, like a single hand only has limited capacity to hold any object. But when you look at two hands, they'll be able to the flexibility that exists in that case will be much more different. And the way in which antigens will be presented in that case will also be extremely different. So that's also the reason for the way in which the antigen or the antigenic peptide is presented. So if you look at this, like for example, if have anything very anyway, if you look at this cleft, if I have to present this peptide and there is limited space, it will get scrunched up. Versus if I have two hands, I'll be able to stretch it out and present it a lot differently compared to MHC1. So that is the difference between those two. And for the peptide motifs that are involved in binding to the MHC molecules, the anchor residues for class 1 MHCs are at both ends of the peptide. They're generally hydrophobic C-terminal anchors. Versus for class 2 MHC molecules, they're distributed along the length of the peptide. If you remember, he had shown that there are point of contacts between the antigenic peptide and the MHC at multiple sites, which is also what helps in that sort of laid down fashion of presenting the peptide. The nature of the bound peptide for class 1 MHCs have an extended structure in which both the ends interact with MHC cleft, but middle arches up away from the MHC molecule. Versus for class 1 molecules, the extended structure is held at a constant elevation above the floor of the MHC cleft. So it's more like a sort of, how to explain, aligned elevation that is maintained. So this shows a space filling model for MHC class 1 and 2 molecules with bound peptides. The first figure is for a space filling model of human class 1 molecule HLA-A2 with a peptide from HIV reverse transcriptase. Amino acids are 309 to 317. So only about 10 residues are here in the binding group. Beta-2 microglobulin in this case is shown in blue. So you see it has no role to play with antigen binding, but it is present as part of the entire MHC molecule. On the other hand, residues above the peptide are from alpha-1 domain and those below are from alpha-2. This is a human class 2 MHC molecule, HLA-DR1, with the DR alpha chain shown in white and DR. You don't need to get into this now because we have not discussed, but just in case you are getting a little confused, these are DR alpha, DR beta chains that they have mentioned here. But yeah, these are DR alpha and DR beta chains that are present and they are shown in white and blue respectively. The peptide here is from influenza hemagglutinin. Hemagglutinin is an influenza. Let's think of it as the main protein present within influenza virus, which is able to generate the extensive diversity that is present for the virus and which is basically what causes the disease. And therefore, in response to that, we are able to trigger an immune response and are able to fight off influenza eventually. Actually, influenza virus is it's a wonderful topic. If you're interested, I will bring it up in the next class, but yeah, we get distracted anyway. So it's a wonderful topic. Please look at it yourself because if you're familiar with the idea of vaccines, we have not discussed it much, but if you're familiar with the idea of vaccines, you will know that influenza vaccine is launched almost every other year or almost every year, and still there is no cure to it. That is because of the arrangement of the influenza genome. Influenza does not have one long stretch of DNA or RNA. Like it's a virus, so it will either have a viral or either have a DNA or an RNA genome. So in case of influenza, it's not a retrovirus, it has a DNA genome, but its genome is not continuous. It has fragmented genomes, right? Much comparable to that of the immunoglobulin gene arrangement. So because they are present in gene segments, you can get a lot of combinations, which is a huge contributor to antibody diversity. Similarly, for influenza, its genome is fragmented as a result of which what it presents on its surface. So what viruses, if you're not already familiar with virology, what viruses basically do is that they have, they're like, think of them as a box, an empty box with genome inside. And that box is decorated with certain entities on the outside. With those surface entities, with those surface epitopes, they are able to bind to host cells, get inside, push the genome inside, and the rest of the box is just thrown away. Then that genome duplicates and that causes havoc within the system. They can, some viruses are capable of killing people. For example, we know about coronavirus. Now we are hearing about monkeypox virus. Previously, it used to be smallpox and a lot of other cases. Not all of them are lethal, but yeah, a large part of them severely affect the host system because that box is just, it's useless. It's the genome which is capable of completely hijacking the host machinery and taking over. And because it operates in this way, viral response is not always very immediate. It's a little difficult to tackle. It cannot be tackled by common medicines. Like you cannot take a antibiotic which will kill the bacterial cell. You cannot take an antifungal which will kill the fungal system. It's a virus. It's not even a living organism. It's just a genome which is circulating around, producing proteins and just utilizing your machinery for its own job. And from there, it is creating new and new viruses, which it will keep on releasing, which can even exit your system, infect other people, and it's a never-ending thing. So that's why viruses are extremely smart. Among them, influenza virus it's mind-blowing. You can read about it from, I think Voigt and Voigt has it in details. So what it does is every like sometime upon some gap, every time you come up with a vaccine, the virus keeps on modifying itself. It keeps taking combinations of its genome and presenting different proteins on its surface. So once your immune system has been conditioned with a vaccine against the influenza virus, the influenza virus changes its decoration and now you can no longer recognize it. So whatever vaccine you had taken up, it basically becomes useless. Something similar to that was also observed during COVID, that new strains, or strains was the more generic word which is used. The classification for viruses is actually different. There are different terminologies. There are serotypes, strains, etc. We don't need to get into it. But yeah, the virus kept on modifying itself every time we came up with a vaccine. So that's how that works. I got distracted from it because it's just to understand that just having a vaccine is not always a one-shot solution to any problem. Pathogens are also smart. They will also modify themselves and they'll also try to tackle it. So you need to keep up with it. Now, coming back to this topic, so we can wrap up today's class, is that the types of anchor residues which are present are different for MHC1 and MHC2. So here you can see the anchor residues from non-americ peptides eluted from the two class 1 MHC molecules. You might think of the fact that how is this at all decoded, that what is the sequence which is what is the stretch of amino acids, what are the anchor residues, how do you figure that out? So basically you can isolate proteins. There are ways of doing that. You can isolate specific proteins. How do you do that in a lab? You today, considering the resources that we have in the current scenario, you can have very specific antibodies or very specific proteins, and you can perform an experiment known as immunoprecipitation. So what you can do is you can take cells, human cells in this case, a large number, say 1 million cells, for example, and you can lyse those cells, rupture them. You can take the protein fraction of those cells. That can be very easily done. We do that regularly. You just need to spin cells. You need to use a certain buffer known as REPA buffer. Lyse cells, only take the protein out from it, protein extract from it. Use that protein extract for your immunoprecipitation experiment. So for immunoprecipitation, you will take an antibody specific to either MHC1 or MHC2. They need to be very unique antibodies so that you do not get any false results. And you remove or you separate out only class 1 MHC proteins from the entire protein fraction. Now, from that MHC fraction, you will get this population of only MHCs from those 1 million cells. From that, you can specifically take out the protein that was present there, and then you can study it. It's slightly difficult to do, but yeah, it was done years ago in 1994. It was done. It must have been a different technique. I have not studied that paper. You can refer to it for your own understanding. Even I can look into it. But this is how you can do it in today's time. So what was identified was that there are some conserved residues and some consensus residues. Conserved residues means that these are always present in that case. For example, for class 1, they found glycine and proline to be very, very abundant in positions 2 and 3. And at position now 9, they found some consensus, which means that they are usually of similar nature. Whereas for class 2 MHCs, they found that they almost always have sorry, they have H2D and H2K have been observed. They're just different representations. They found tyrosines to be present, and here they found that there can either be isoleucine or valine, etc. So these are examples of anchor residues that can be found from class 1 MHC molecules. Class 2 has not been shown here. So you can find some specific residues to be present, and they are important for the proper recognition and proper binding of the antigens, and therefore they help in effectively finding and maintaining a certain orientation of peptides when before they are presented to the correct cells. Now, what happens after that has already been discussed by Sir, that there is recognition by the co-receptors CD4 and CD8, which strengthens that interaction and therefore helps in proper recognition, and then calls for TCA-TH cells, and then it elicits a whole elaborate uh, hyperactivated immune response, and then that particular pathogen carrying that antigen will be eliminated eventually. So with this, we come to an end of today's class. That is all I had to say. If there are any questions, I will take up that doubt you brought up, Namita, about the antibodies which can cross the placenta. But apart from that, if there are any doubts, please let me know. I'll actually put that up before I upload the slide also, so you can look into that directly from the slide. But if there are any more questions, please let me know. If you need any other approach towards the classes, please let me know that as well. And just trying to explain things based on what I think would be helpful. But yeah, please let me know if any other information is required. Okay, if not, then thank you so much for joining today's class. You will find the lectures uploaded and also the slides. Please come up with more doubts and encourage other students to also join the live interaction sessions, not just mine, but anybody, any of the day's interaction sessions. They should be helpful. But yeah, thank you so much for joining and have a great day. And I hope these sessions are helpful for you.